Welcome to Happy Planet, an impact economy podcast where we speak with entrepreneurs, investors, and thought leaders in search of profit and planetary health. I'm your host, Abigail Carroll. Our first season is about the ocean economy, a world I'm intimately familiar with, and where some of the greatest environmental impacts are being forged from business innovation. In this episode, you'll meet Griffin O'Driscoll. Griffin is the exceptionally young founder and CEO of Organicin Scientific, a biotech startup with the potential to revolutionize how we treat harmful bacteria. The company's mission is to reduce the world's reliance on antibiotics by providing safe and sustainable alternatives that will protect our food supply and our population from disease. When presented with opportunities to launch in the pharmaceutical market, agricultural sector, and beyond, Griffin and his team decided to start their journey with aquaculture, where, just like in agriculture, harmful bacteria present serious threats. From food safety concerns for oyster farmers to mortality issues in shrimp farming, it was fascinating to learn about the solutions presented by their technology. Let's jump in to better understand the nuts and bolts of this biotech and why I think it's such an exciting opportunity. Welcome to the podcast, Griffin.
Hi, Abigail. Thanks for having me.
Let's cut to the chase here. You are 3 years out of college and you are a CEO of a biotech startup that you founded with a former professor and classmate that has global paradigm-shifting potential. First of all, congratulations. And secondly, how did you land here?
Yes. Well, it wasn't completely on purpose, but basically, I studied biochemistry in undergrad at UMass Amherst. Before that, I was a bunch of different majors, some that had to do with science, some that didn't. At one point, it feels like I fell into the lap of it. But like any life science student at any college, especially at UMass Amherst, there's a peer pressure somewhat to get involved in research and actually start gaining some experience, and whether or not you're a medical school student or do you actually want to be an academic professor or work in industry, you get this experience. And so as soon as I transferred over to biochemistry, I was a sophomore, I started doing the cold emailing just about any professor I could see, regardless of what they were actually studying. I just knew I needed to get some experience, get something on my resume. And so I did the cold emailing, and of the 10, 15, 20 people, 20 professors I emailed, only one answered, which was completely okay with me, looking back. And that was Dr. Margaret Riley. And she was studying bacteriocins. These are anti-microbial proteins in the race for preventing and treating bacterial disease. Bacteriocins have a very large role, at least potentially. And that really interests me. So there were, all these other professors were doing all this other research that I didn't really understand, but finding alternatives to antibiotics in a world that, sirens the alarms on antibiotic resistance, even coming from outside of a life science discipline, I very much understood that. And so I was very happy to one, just get into any lab, but two, something that was a bit more meaningful, a bit more visual, and something I felt that could actually have much more of an impact on the world. And it was something other people understood too, which is always great.
That's great. So these bacteriocins are, to us it sounds new, but there's a history. Bacteriocins have a history. It's sort of what's old is new again. Is that correct?
Yep. Fashion is very cyclical, and it seems in this case science. So yeah, the three big pathways of preventing and treating bacterial disease or controlling bacteria are antibiotics, bacteriophages, and bacteriocins. So these are kind of the three most direct approaches. And you've probably heard about antibiotics. I'd be really surprised if you didn't. And you've probably heard of bacteriophages. And I would actually be surprised if you heard of bacteriocins. And so they all started around the same time. So the germ theory, where we actually realized it's not bad air that's causing illnesses, it's actually microbes. It was in like the late 1800s. Right. And then, shortly after 1928 is when Alexander Fleming came across penicillin, discovered it kind of by accident. And that was the first instance of kind of this new age almost of where we can actually start developing treatments to something. I mean, before this, you could get, it's not impossible for you to die from a scrape on a knee or strep throat or something like that. So a lot of things we take for granted today that's easily treatable by antibiotics really wasn't possible prior to their discovery.
Right.
Bacteriophages and bacteriocins were also kind of came about in that same era. They really just got pushed aside thanks to the phenomenal efficacy of antibiotics,
Right.
as well as a big geopolitical story just about World War II coming into play here. A large fire, actually both in Boston and in London. It's a fascinating history. So during World War II, we needed to produce at scale antibiotics, and so that was a big kind of stepping stone in which Fleming might have discovered this antibiotic, but that's not really that useful if we can't give it to a lot of people. And so with the U.S. and the West's need to maintain soldiers, there was a lot of funding put into actually building these large-scale production facilities and kind of figuring out that process. And so, it's very rare you get kind of a whole country's mind to work collectively on something like that. I mean, COVID pandemic is quite literally the probably biggest story there. And so we were able to, that was the start of Big Pharma. So companies like Pfizer, Eli Lilly, that's really where they got their start with antibiotics. So antibiotics were actually the backbone of kind of Big Pharma that we know. Some people love it, some people don't today. Meanwhile, Germany and Russia had their part, right? Each picked either the bacteriophage or the bacteriocin. But ultimately, the war went the same way. The kind of fate of how we treat bacterial disease went. And so the West liked the antibiotic, and it worked really well. And so that's kind of where we put our money and our energy behind. And of course, 1950s came around. That was the golden age of antibiotic discovery. We just kept finding these things one after the other, one after the other, one after the other. And so it was quite literally, dubbed the miracle drug. But eventually resistance caught up. And around the 80s, 90s, it became even more of an issue. And today it's a huge issue. So just over the COVID pandemic, I think antibiotic resistance infections increased like 18% just in the 1 year. Just because we had no idea what COVID was. There were so many people in the hospital. We were just throwing antibiotics away like candy. And so the other piece of this is the understanding of the microbiome. And so that wasn't a thing until the past 20 years or so, and even more so like the past 10 years. We didn't understand that the microbiome really existed, let alone that it had a function, let alone a really, really, really important function. And we're still understanding it. We're still in the very early stages of seeing the implication of the health or the microbial diversity of a microbiome, and that's its impact on our mental health, our physical health, disease causing or immune fighting, right? All these different things. It can be proactive, it can be reactive. And where we are today, in which we are trying to find alternatives, what's really causing that is this antibiotic resistance that's occurring, as well as our new gained understanding of microbiomes and knowing that we don't want to disrupt them. And that's what antibiotics do. They disrupt them. They work broad-acting. They work indiscriminately. They kill all types of bacteria. But these new approaches, bacteriocins, bacteriophages, are very, very specific. And that is a tool we can use to still get this disease-fighting mechanism, but without the collateral damage and the effect on the microbiome.
Right. So let's dig into the bacteriocin just a little bit. The bacteriocin is created by one bacteria to stave off another bacteria. Is that correct?
Yes. Yeah, they've existed in nature for billions of years. And so they've kind of evolved to, bacteria, when fighting with other bacteria over resources, the bacteriocins are expressed. And they're expressed to kill that other bacteria so that its kind of species can survive and it can get those resources, those nutrients. And so it's very much the weapon of choice, I like to say, within kind of bacterial warfare.
Yeah.
And this is one of the big reasons of why we like them, why they're safer, right? They're non-toxic, and they're more narrow-acting, is because they only care about their exact microbial community, and they only care about killing bacteria. So there's not a lot of off-target damage, both in terms of other bacteria species as well as things like human cells, right? It only exists to kill that other bacteria so it can survive.
Amazing. So if you're trying to target bacteria A, you as Organicin, you're going to be looking at bacteria B's bacteriocin that's actually targeting bacteria A. Did I say that right?
Yeah, that's right. There's two parts to our company. There's our platform. So really what Organicin does today is we're a drug discovery company seeking to commercialize bacteriocins. And we do that through our discovery platform. And there's two pieces to that. There's the predictive modeling, and then there's the phenotypic screening. And so the screening involves taking a petri dish, streaking a bacteria strain on it, and then dropping a bunch of different bacteria strains, like with like a little dot, and seeing if it kills. So visually, I mean, we end up measuring it, but you visually see if it killed or not. And we repeat that at a very large scale. So we do that all day, and there's a lot of technology now where you don't actually even need humans to do it. We of course still do because we're an early stage startup, but yes. And so it's very much like which bacteria killed which bacteria, and from there you can we confirm that it was a bacteriocin. It almost always is, but as simple as that sounds, there's 1 trillion species of bacteria, and so you can spend your whole life doing this, and you won't even come close to finding all the bacteriocins, let alone just because something produces a bacteriocin doesn't mean it kills it in the way you want to. Coming at this from a business perspective, just because it kills on the plate doesn't mean anything. Will that bacteriocin still work? Even if there's salmonella in chicken or salmonella in humans, will that bacteriocin work in that physiological environment the same way? Probably not. And then you take into account, aquaculture, will it work in the ocean? There's a lot of different environmental factors that an academic lab really wouldn't take into account, but what we do. And so that shows up in our predictive model. And this is really kind of the big differentiation. So we start with that business challenge and try to take into account those parameters so that when we go and do this screening, we're very thoughtful about which of the 1 trillion bacteria we select to kind of what we call we're kind of simulating fitness or competition, right? So we're really putting these against each other and making them fight and seeing if they produce the right bacteriocin. And so that's what we do at a very large scale. And the predictive model is what allows us to not have to look all across the globe and at all these bacteria, but instead pick the right bacteria to fight against each other to get the bacteriocin that we want that ultimately will be a useful product and that'll still work in the environment that we intended to.
You've got this incredible technology. You have a platform. You're discovering different bacteriocins and applications for them. Where are you starting? What's your go-to here?
Yes, our go-to-market. It's quite unique, and it's not super consistent, which is always fun to communicate to investors. We've came across aquaculture. Why not pharma is a very passionate question I get often. And that's because, at least initially, we were 21 years old, and nobody would take us seriously enough to think we could bring a drug through the FDA. So there's that.
Said with the best humility.
Okay, we won't do that. So we actually started in treating fire blight in apples, and we found a lot of really, we still to this day have a cocktail of bacteriocins that can kill 99% of the pathogens causing fire blight with, I think it's only killing 6% of the probiotic bacteria, the plant growth promoting rhizobacteria. Phenomenal product that I hope one day we can actually come to market. But agriculture is, I think it's one of the oldest industries of society, right? That's the reason we stopped being hunters and gatherers and were able to actually develop society. So it's very old. So people are very stuck in their ways. And the using something innovative they're just not as open to innovation. But we came across aquaculture, a big thanks to Blue Swell, India kind of being located where we were. So we were based in Boston, in Amherst, right on the coast. A lot of aquaculture, or a lot of seafood, big hub of seafood industry.
New England.
Yeah, of course. Yeah. And aquaculture is a new industry. It's, we recognize the need for more protein. We can't catch it from our oceans anymore because we've already depleted those stocks. And so we have to grow it, whether that's, on the shoreline, mariculture, or actually land-based. And oysters were a very local, kind of one of the biggest aquaculture, so actually farming seafood, species geographically. So, Massachusetts is big for that. Maine is big for that. Prince Edward Islands, you got Canada as well. And so aquaculture being this very young industry, they're much more open to innovation. And With oysters specifically, so that's, if you can't already tell, that's where our first product is being oriented to. Our first application is oysters. And so there's a pathogen called Vibrio perihemolyticus and Vibrio vulnificus. Vulnificus is much, much nastier. And it's a pathogen that absolutely loves warm waters. And so it has always been an issue in the Gulf. And if you've ever heard of someone getting sick from oysters, it's probably one of these two pathogens. However, climate change has warmed our oceans, and now we are getting this pathogen in these parts where we haven't typically seen them, like Maine, where there's cold water. Again, the Pacific has always had this issue, but even more so now. So like outside Washington.
Yeah, it's just warming everywhere. They're starting to see it here in Europe too. So
we were made aware of this kind of market opportunity, and Dr. Riley was like, Vibrio, oh, we can do that. That's no problem. And so we kind of pulled from our library of candidates and started to get to work. And surely enough, we could kill it time and time after again on the Petri dish. As we got closer to the actual farmers and the wholesalers and understanding the value chain there, we saw the market opportunity was attractive enough to go for it. And so that's what we're doing today. We are currently testing this in vivo. So we've done some preliminary trials testing the efficacy of these proteins in oysters. It was successful, and we are now doing that.
And they haven't killed the oysters.
Of course not. So these are essentially designed by bacteria to kill other bacteria. So they don't harm anything else besides bacteria. And so there's no damage. There's no reason for it to affect the taste or texture of the oyster either. We'll have to confirm that, of course. But I mean, that's the beauty of these things being proteins. They're so small. They're highly. There's such high efficacy that you don't need a lot. And so there's from a lot of different angles. They're really perfect for this type of application.
That's amazing. So the oysters, they'll filter through this? How do the oysters relate to the bacteriocin?
Yeah, this is where the go-to-market gets a bit interesting. So the oyster, we need a closed system to actually give them to them. We're not going to just dump this in the ocean. It'd be safe, but it would be extremely expensive because we would need a lot of bacteriocin. So we need a closed system. And oyster wholesalers have that system. It's called a wet storage tank. They use them today for both inventory control as well as to essentially purge, so that the oysters can kind of filter the water and decrease that bacterial load of Vibrio. And so that's a great point for us to deliver our solution. And we've proven through our preliminary studies, they take it up, they eat it, and it goes into their organs. They filter it, and that's how it ends up getting delivered to the right places kind of internally.
That's amazing. So you're starting with oysters, and what does the future hold for you guys?
So our platform is industry agnostic, right? As long as it's a bacteria, we can kill it, and that opens up an unbelievably large amount of markets to a point where it's overwhelming, right? There's human health, of course. There's like skincare, but we're sticking with the aquaculture route for now. As I mentioned, they're just the industry is much more open to innovation. We are using a ton of antibiotics in agriculture, but for a lot of different reasons, that's just not being allowed in aquaculture. Maybe because the bar is a lot higher. People are demanding it a lot more, so consumers. But the whole idea of aquaculture is it's this kind of untouched, right? It's seafood, it's super healthy, and so we're not using antibiotics. And then in other countries, it's just outright being banned. And so our next product is less a food safety concern. So that oyster issue is right. That pathogen doesn't harm oysters, but it harms humans when they eat it, and people get extremely sick and they die from it. I've started to hear those stories of people coming up to me and saying, whoever was sick, I really appreciate what you're doing. And that's just another point for why we can't quit, because now it's starting to get to, you hear these personal stories of, wow, okay, people really need this.
I mean, as an former oyster farmer, I lived in absolute fear of giving somebody a Vibrio. It was terrifying.
Mm-hmm. The number of people that get severely ill or die from it isn't huge, but sometimes investors don't look at that favorably because they think it's a small market size. But once too many in the book. But anyways, so our next market is actually shrimp, and so this is a animal health endeavor. And so actually, this pathogen, it's the same pathogen, causes mortality in shrimp production. And so we're going to be taking, again, likely a different bacteriocin, or maybe a cocktail of the one we're using for oysters and some other ones, but delivering it via the feed. So making a feed additive and giving it to shrimp so that the bacteria can get delivered to their or the pancreas, and preventing that pathogen from kind of taking hold. And this is a very large market. So shrimp, everyone in the world loves shrimp. It's one of the most valuable species in terms of aquaculture. And this one disease, it's called acute hypopancreatic necrosis disease. They abbreviate it as APNID. It's caused by this Vibrio. It wipes out farms. I think in 2011, it just traveled through the world, and countries like Thailand are still trying to get their production levels back up to that kind of 2011 era production numbers. And so there's a very large need for ways to kill it. We used to use antibiotics, but again, the regulatory environment now, less even the consumer demanding it, but like in India, they just outright ban antibiotic use because they need to stay competitive with all the other markets trying to sell into the U.S., the EU, China, Japan. And the U.S. and EU increasingly are testing for antibiotic residues and sending these shipments back.
Step back. Wow.
There's just so much demand from like governments, from the farmers, from all these stakeholders. We need new alternatives.
It's interesting that there is this different standard between land farming and aquaculture, but let's hope that sets the bar higher for all foods. But so the shrimp farming, is this all in mangroves or is this all circulating systems in India, for example?
So it's all pond farming. There are these high-tech recirculating aquaculture system, RAS. Those are more, again, EU, US, but and they have some issues there too. They won't really tell you that, but these bacterial pathogens still exist. It's hard to prevent microbes from entering places. Sure. They're so small. But where we're focused and where majority of shrimp production comes in the world are these ponds. So they're usually close to the coast, but they're very much kind of land-based. And that's, Southeast Asia, Central Latin America, and China are the big producing countries there.
We'll be right back after a short break. For a blue economy to thrive, people need to use more sustainable products. But which products? And will consumers actually adopt them? Innovators like you are hustling to figure this out. Spark number 9 can tell you if there's demand for your product. Spark markets your product before it's built using online advertising so you launch smarter. Have a big idea? Vet it with Spark before you build. Visit Spark online at www.sparkno9.com or find them on LinkedIn. Blue Trace offers the easiest and most powerful traceability solution for shellfish harvesters, growers, distributors, and dealers. Their tagging and distributor apps enable organizations big and small to optimize their activities, comply with regulations, and keep up with their inventory. For more information, visit their website, blue-trace.com. Welcome back to Happy Planet.
So I want to get back to you for a moment because you graduate from college in 2019 and you've been in this exciting lab environment, but then you head off to business school. And I'm wondering when you had sort of the aha moment that we could really make a business here. This lab work could actually be commercialized.
One of the reasons, like why I wanted to join the lab was, I saw it from the get-go that this was something usable because you almost grow up, if you don't live in a cave, you hear about antibiotic resistance. So I knew it was top of mind, and only over the years has it gotten louder and the calls for alternatives louder. But when we wanted to actually do something about it and spin it out of the lab, make a company, it was my senior year of undergrad, and we were just at our weekly lab meeting, and the business school had a pitch competition. Dr. Riley asked any of us if we wanted to do something. She's had people in the past try and actually won. So they've won, I think it was a company called Bacteriocins, and won about $30,000. And so she asked us, and I raised my hand. Well, I looked around first, and no one else was doing anything. So I raised my hand. And then I pulled in Matt because Matt was also in my lab, and he was my friend at school. So I was like, I'm not doing this alone. And we made an application. We submitted it on that Sunday. It was a terrible idea. And that didn't deter us. And we got a lot of good feedback. And that is the best part about the startup community is the very honest feedback. Usually it's not super negative or discouraging. It's just very constructive. And so we just went to work iterating it over the years. And that's what we did. I went to business school. I went to Boston University. And it was there. I mean, I never gave this up. Matt was younger than me, so he was still in school. And we just kept building and going and pushed in a lot in some really good directions. And why we never gave it up was no one ever said, like, this is a bad idea. Like, it was always like, you need to do this, you need to get this, you need to, show this. But it wasn't like, this is a stupid idea, you should never do this.
Right. There was never a hard stop.
Yeah. It was like, you could just feel it. Like, if we, the only reason we could fail is if we quit. So we can't quit.
Right.
And then eventually, I graduated from business school. I went into consulting for pharmaceutical market access. And then there was the Blue Swell incubator. So this was a Boston-based incubator focused on blue tech companies, partnership with the New England Aquarium and a community called See Ahead. And that was, we actually applied to there once, didn't get in, but we met some of the reviewers who, again, were like, yeah, you guys are too early. You don't have, the metrics, you don't have the milestones, but it's a great idea. And we'd love for you to work with you and see if you can get in next year. And that's exactly what we did. We did get in that next year, which gave enough funding for me to go full time, and that was kind of the start of this journey. And that was coming up on just 1 year ago, and so it really, that's why I said at the beginning, it kind of felt like I just fell into it. It just kind of snowballed. I said yes in that lab meeting, and then I just never found a good reason to stop going.
That's as good a reason as any other to keep going. At one point, there's no roadblocks, and then hopefully you get to another point where you're actually getting pulled along. It's a little bit of a different momentum. So you're bringing this research from a lab into a commercial sphere, and I think that's a really big challenge for a lot of people. Why do you think some things just get stuck in labs?
There's technical risk, there's market risk. And, the good thing about something like, say, deep tech, or if you're working in an academic lab, you're trying to spin it out, it's probably pretty technical. But whenever you're doing something technical, the market risk usually isn't, that's the trade-off. Like, we're talking about, right, developing treatments for diseases that kill people. So someone's going to buy that. But can you actually make it? Can you actually make that drug? So that's where, like, the technical risk comes into play. And I think a lot of people just don't understand that kind of the other side of things of, again, you can find a bacteriocin, but can you find the right one that actually works in the application you need? So can you actually take this research that sure could be game-changing? Yeah. Can you make it into a product that actually has, like, unit economics that makes sense and is actually works in the way the customer needs it to? And so there's missing that piece, and there's a lot of different support systems, I guess, here. And so, like, the NSF I-Corps program. So that's like working with academic professors. What they do is they do customer discovery. So they give them some money and they force them to call, X number of people, 20, 50, 100, to get that customer impact as a way to kind of get it in their heads that the customer is the only way this thing is going to come out of the lab. And then, of course, startup accelerators and incubators have a role to play here. But I think there's just kind of, it's hard to be a PhD scientist and like an MBA at the same time.
Right. It's a different culture. Yeah. You're the great model, maybe. Students coming through and finding some really exciting platform and spinning it off. But I think the other thing that you mentioned was the accelerators, like having more of these sort of business and entrepreneurial and pitch events, and having that new culture inside the universities can actually make a big difference too. You've been in a number of accelerators, correct?
Yes. Yep. So there's a little bit of a difference between incubators and accelerators, but yeah, these startup communities and programs that typically give you funding and a lot of networking and advice and mentoring. And so we did Blue Swell just last year, and then shortly after, and where we are today, is Techstars. Techstars is an accelerator. Typically, accelerators are a little bit later stage, and so there's a higher bar to clear. But yeah, they are an amazing part of the startup community that, again, these are pretty new ideas. So like Blue Swell's on their third cohort. I think I'm I think this Techstars accelerator, we're one of the early, maybe the eighth, maybe the fifth cohort. But regardless, these are very new.
Yeah.
And it's just, You can go to, like, I went to business school. No one tells you how to really start a startup. I wasn't an entrepreneurship major, so I don't know how that looks. But even like one of my favorite things right now is I'm very focused on sales and trying to show commercial traction, which is hard to do when you're a biotech trying to develop a drug that that you're still researching. And I'm like, no one ever taught me sales in business school. And so there's this. It's essentially school for how to build a startup, and you meet a lot of people who have been there and done that. A lot of subject matter experts, a lot of people that have just successfully ran businesses, and from every angle, legal, again, sales, marketing, operations, finance, and you just an unbelievable network. And the word accelerate is so true. You're just able to completely 100% focus in a very inspiring environment, shoulder to shoulder with a bunch of other entrepreneurs doing the same thing in likely non-competing markets. And so you're very, it's very helpful. And I think it's one of the most important pillars of entrepreneurship.
It's interesting because they're popping up in all over the country. I'm recording this from Paris right now, and there's tons of accelerators here. And sometimes I wonder if it's hard to make progress in the company while you're doing an accelerator.
It shouldn't be, right? The purpose of it should be to improve your business. And so, like, we're very clear about that here. That what I like about this program is it's very metric oriented. And so this whole idea, if you're not measuring, you're not. How do you know you're even doing something? If a tree falls in the forest, how did you know? So you have to be measuring. And so it's very much like, what are you doing week by week that is improving your business? And that can differ, and it does differ week by week. But at the end of the day, they're not, these programs, these mentors, the network, they're not you. They're not running the company. And there's a bunch of other things they got going on in their life. So it's up to you to prioritize your time and make sure you are doing the things that matter and that do actually progress forward. And sometimes that doesn't show up in a metric. And so there's, metrics aren't perfect. So ultimately it is up to the entrepreneur to kind of make sure that the fate of their company is going in the right direction.
Right. You're there to win. If you drop the ball, it's, you suffer the consequences. So speaking of investment and fundraising, I know you've made a round and you're going to probably do another round soon, pending some results. Where are you with all of that?
So Techstars has been kind to us. Minneapolis has been kind to us. And we, along with some angels back in Boston, were able to put together a pre-seed round that gave us enough funding to hit some pretty key milestones. And those are this in vivo data I talked about earlier. So showing that, that's a big de-risking event. It's not easy, but it's not super hard to show results on a petri dish. But when you introduce the complexities of life and actually on these organisms themselves, the oysters, and you can show efficacy there, that's meaningful. And so we're doing that right now. That's very top of mind. And then the other things we're trying to do is just show that commercial traction. And really what that means is getting that good data so we can go to our customers, which we actually do a out-licensing model. So, for oysters, it's very direct because there are no other people doing what we're doing. But in shrimp and in this kind of animal health sphere, there are very, very large companies, feed manufacturers that ultimately are going to be our customers. We're going to out-license our products to them. And so showing that commercial traction, getting letters of intent, as well as a techno-economic analysis. So understanding our production costs and how that changes over time, how that scales. And that's something that I've loved to learn about over the past couple of months of like just getting it into my head that it doesn't matter if you have something good. This goes back to kind of that whole antibiotic discovery story. You have to scale it. If you can't scale it, it's almost meaningless. And for some of these large animal, these feed companies, it's like just because you have good data, if I can't get it consistently at the scale I need for my customers, then we're not going to do anything. And so being able to understand that, how that production cycle looks and how it scales and the volumes needed and the cost is going to be really important to be able to say, like, hey, this works in the organisms. We can do it cheaply. And then that'll allow us to hopefully raise our next round. So yeah, we're hoping to raise a $3 million seed round in January.
Well, that's great. And in 5 years, where is this going to be?
5 years, we would have raised our Series A. 5 years, we'll be like, have our hands in all sorts of things. So by then we would have had products for salmon and tilapia. We would have gotten into agriculture. So chicken, swine, maybe cattle, and ideally have started to look into the human side of things. So not even close to anything resembling a product, but starting that early discovery work of, maybe what pathogens would we want to kill, and can we get some early in vitro data on that? But really just starting to build out the company, build out our processes, and I shortly touched upon the predictive model. That is in a very nascent stage right now, but like the more data we get, every time we search for new bacteriocins, we get more data, and I just can't wait for the day where like we just have this massive database of that will just push forward our understanding of bacteriocins, and then we're in a spot where like discovery is just super, super efficient, in which we understand these things so well that we can find that bacteriocin that does kill that human pathogen, like I won't say easily, but quickly. And so I look forward to kind of building out the platform technology side of the company, just as much as like the revenue generating products.
Well, thank God Dr. Riley invited you into her lab.
Yeah, I should buy her dinner one day.
What advice would you give to other entrepreneurs who are looking at you and dreaming of becoming like you?
Yeah, I owe so much back to the startup community. So the first step is to just like dive into that community. And if you're like younger, like if you have a university email address, you can essentially talk to anyone you want in the world, because most people just want to help. And like the amount of innocence you have as a college student saying, I want to learn about this, I don't know why anyone would turn you down. So use that to your advantage and just start talking to people, get that feedback, and really just go for it. Like shoot for failure in a way. Find out what doesn't work so you can then focus on what does work. The faster you can disprove things, the faster you can get to the point where you can focus on the thing that does work.
Spoken like a true scientist.
Talking to Griffin, I gleaned a lot of valuable information about the future of biotech, the long road from a lab to product launch, and the mechanics of bacteriocins. But my biggest takeaway was that Organicin is much more than just a company. It has a mission that stands to change the lives of farmers, which I think we all can agree is key to feeding a growing planet. Thank you for listening. Please follow the Happy Planet podcast wherever you listen and leave us a rating and review. It really does help new listeners discover the show. Happy Planet was reported and hosted by me, Abigail Carroll. I am the executive producer. The talented Dylan Hoyer is our producer and editor. Our theme music was created by composer George Brandel Egloff. Learn more about my work and get in touch by visiting happyplanetcapital.com.